Okay, my friends, this is a 3-foot wide human hand. It's been DNA certified. Now I'm going to explain to you what you're seeing here because it's very, very unbelievable. I mean, I could see all of the articulation in the skin and everything. You can't see it until you get down with a better way to see it. But if you see this silver stuff, you see this silvery looking stuff right here and over here. You see that? That's not snow or some reason it's there. That is silicon. That is actually silicon. Your grip skin is dense with silicon. It is grippy, it's tough, it's scrappy. Silicon is tough. Now, and I was finding these layers here now as I was taking it out of the ground, separating and so forth. Now, and like I say, this thing's huge. Huge. Now, this, if you took your hand, the hand here, let me turn this up. It's your hand laying backwards like this. All right, and if you look, you have one of these things, a tendon running down there, and it runs right down right here. This is right here. This is right over here. All right, so it's gigantic. Now this is the pad on the back of your hand. That's this pad right here. Here. It's like that reddish looking color. It's like, it almost looks like it's puttied running down here. You see it? Now I have found many of the fingers and so forth, and I have them over here, and we're going to go through them and look at the chemistry of how the tendons attach and the knuckles and so forth, because I found a bunch of stuff since then. Now, this is 7 years ago. Nobody's interested. It's still laying out there. I think it's all deteriorated by now, but nobody paid any attention. So I am going back at this because I will not let this die. This is DNA certified. There is absolutely no reason for people to deny this. This is just tragic. It's tragic. I'm telling you, if I was a student, I would be appalled. I'm appalled, but I'm not a student. But everybody should be appalled. It's just terrible, and especially if you're paying somebody to teach you, and you think they're going out and figuring out what's right. You got another thing coming. They're reading a book to you, my friend. Now, I have 57. I think I had like 58,000. People that are very involved in this, and they send me things in droves all day long. I'm having a very hard time keeping up with it. Now, this came in from Gary Evans, a few years ago, I think, and Gary found this in a salt mud flat, and that is the key. That is the key to the preservation of mud fossils. I have created my own mud fossils, and I understand the process. And the process is salt water of approximately the pH of the body, and then all of the soft tissues are very happy in that condition if they stay submerged in that particular condition with no oxygen, just floating around inside. So what happens to them? They stay wet. What happens inside? They stay just like they were before they were dead. All right, this is a lung, and Gary Evans found this over in England in a salt mud flat. So it was in the salt water. You see the little pebbles all over here? That's from lungs. Lungs have all of these different transition metals in the lung tissues, and internally here, it hardly even deteriorated. Now the black is from the blood that is going into the lungs to be reoxygenated, and of course the red is the reoxygenated blood, and it transfers over. Now let's see, this is as it was drying out and it started to fall apart. Now let's see what happened as it went forward. Alright, so then Gary put a little water on it. I said, let's take a look at it, get it wet so we can look and see all the different colors. Sure enough, it's just gushing blood out of here, and of course that is the passageways into the little holes that are in the lungs where all of the oxygen transfers through carboxylation. It's carboxylation. And I'm going to show you something what's in a lung, because this is another thing they don't understand. It's not just air goes in there and it comes back out with carbon dioxide. Absolutely not. All right, this is a lung from, I don't know what kind of creature it was from. Hold on, let me get this light over here. All right. Now, look at this carefully. Look at what you see there. You see these crystals? Let me get a little pointer. Hold on. All right. You see these crystals here, these different colored crystals? And you see how they're all cracked up and a little fractured looking? And you see how there's all these cracks and crevices in between those crystals? Well, this is a lung. All right, it's the same as the lung I just showed. Well, it's not the same lung, but it's a lung, and it transfers chemistry in and out of your body. You see the different colors? Those different colors are from different transition metals. And what do? They transfer different molecules. You have to have those transition molecules in your body, the transition metals. They carry everything around. The blood passes by these pockets that are coated with these transition metals, and it makes a molecular transfer. So the blood carries carbonic acids. Carbonic acids. That means they want to move their chemistry to a different condition. They come up and they push it right through from these transition metals because of these different particular molecules that are in there, and you can see them because of the colors. And I can prove that, and I can prove your body needs these things in the correct places by a heart that has opalized. Let's take a look at that. All right, I'm sorry. This is a couple weeks after he took it out of the ground, and it was gone. You let it dry out, and I'm good with letting things progress to find out what happens. They put these things in jars, and then people look at them and say, "Oh, look at that." Well, that's not the way you do research. What happened to it? If it dries out, it turns like this. That turns into mud. He puts that in water, mud. This is a human heart. You can't tell any specific things about the tissues there. They all look like they're brown, sort of brown, yellowy looking things. Well, I'm going to tell you something. These heartstrings, these ventricle walls, the cardiac muscle, the aorta, all have exact chemistry. And if you can't see that and you don't understand what you're looking at, I have no help for you. Those are the walls, and these are the little heartstrings, and these are the aorta and so forth, and all of the different articulation of the heart is obvious. It's not just one big brown mass like the other thing was. What else is here? You see that right there? That's blood. Obviously, there's blood in the middle of the heart. Well, this is where it settled. It was probably laying upside down like this. The red blood associated to the top. This is where all the transition metals have collected. You see them, all these different colors? Those are the transition metals. If you don't have those in your body, you will be sick. And there is almost 100 of them. Hold on. There's this right here. You see that? You see where the blue is here? All of this blue. All that blue, and I think up to like 84. There's a certain point where some of these you won't have. They're sort of synthetic molecules. But the transition metals are the ones that you have to move things around your body. Nobody knows about them. Nobody cares about them. They think they're just trace elements, that they don't have any reason to be there. They are there to transfer things with ligands. Every single color here you see is a different molecule. And let me show you something. Now, these are the transition metals. These are the things that you just saw in that heart. And if they're not there in the right places, in the right quantities, at the right time, the guy's going to be sick. Case closed. Absolutely. If it should have been this color and it was this color, you got a problem. Now, you should be able to tell in the blood what molecules are there in the transition metals, and if they're not there, or if they're in too much, there's a problem. And if they're not there or they're in too much, it's usually related to an enzyme has not been able to work with them. And if the enzyme is not there to work with them, or there's an issue with that enzyme, it's usually related to the bacteria that's in your gut, because the bacteria in your gut make the enzymes based on what your body needs. I'm telling you something, your body actually talks to bacteria, identical to you ordering around people in a chemical factory. and you say, hey, Charlie, go get some carbon, that carbon stuff over there, and bring it down to the guy. He's going to add it to some transition metals to go around, and go pick up some of the stuff that's laying around. The guy's getting sore, he's stiff, he's scratching into his membrane. He got all kinds of problems. You know, he hasn't had any of that stuff in a month. Thank God we got it in here. Go get it out there as quick as you can. 24 hours later, the guy says, holy smokes, I feel all right. I'm feeling better. And I'm going to tell you, that's a fact, because I've been working with Jody, who was a friend of mine, and then I'm working with her. I just said, you know, I use castor oil on my sore parts and my aches and pains and everything. She had terrible RA. Now, she's been a year. No treatments whatsoever, using castor oil every day, well, when she has trouble, and using it, you know, on a regular basis, continuously. And I do too. And I have no problems with that either. Now, what was the result? No, she's not taking any drugs. Before, she had to lay in bed. Literally, she couldn't work. She couldn't. She told me there was many days she couldn't lift a pencil. She couldn't. She literally was an invalid, and her hands were swelling up. People were saying, "Oh, your hands look terrible," and they do. They're eating through the membranes because of this mucus stuff. Now, so what does she do? She puts castor oil on her hands, and originally she was wrapping them up in plastic wrap, which is what I said, you know, worked for me on my ankle, because I had the same thing. My ankle caused me trouble. I was starting to walk sideways. It was so sore. And that went on for years. And it was getting worse and worse and worse. And I said, well, I'm going to just soak it in the castor oil. And I did literally for a week. And it's been fine. Now, that was 5 years ago. And it's starting to come back now, because I haven't done it for 5 years. That's the problem. You get away from it. I think it has something to do with repairing the membrane and possibly helping out the tendon anchors, the tendon anthesis points, because they are loaded with transition metals, loaded with them. And I can see them in my mud fossils. Totally different colors where the tendons anchor. Hold on, I'll show you. And that, you see those different colors right there? See these different colored spots? I hope you can see that. That is not just accidental dirt or something. That is the actual chemistry of where these tendons implant. That is the apical tuft, and they stick in all over the place. So it's not just right at the apical tuft, but this. Hold on, I gotta find it. All right, any doctor will, or anybody that knows anatomy well, knows that at the very tip of every finger there's this little attachment. It's called an apical tuft, and it has all these little gnarly holes all in it. You see these holes? That's right underneath. That's right underneath here. This thing is here. That little ball right there is a tendon ball. You see it? That one didn't break off. All the other ones got pulled out. But that little ball will be right there. And this tuft is right here. And then it's down inside, of course. And then it anchors, all these balls anchor around it and hold your finger so that it can move here and there. Now, let me show you the anatomical. You know, before I do that, though, let me just show you. This is one of the knuckles that came off of it. That's the bone ball. And that, see this? That's flesh. And that is the blood. I mean, that's iron from the blood. And that is the tendon assembly that comes down and would have gone over that bone ball. And if you can see here, that's actually the webbing of the muscle that wraps around the other side of this bone ball to hold, you know, I mean, you understand what it does. Now, that runs down to the tendon on the side. And I'll show all this in the, I have a couple of videos on this. And all of these things were from the same thing. But it was in two areas of you know, erosion. You can see how perfect this was. And I have other ones that are literally perfect. I mean, that is actually like a flawless fingertip. And this one eroded much more, but you can see the distal phalanges in there. And right at the tip of these distal phalanges sits this. Now, that's where the arterial side blew out. The The vein side doesn't blow out. See, the vein side is like this, no holes. This one has the holes. Hope I'm holding this in the right position, you can see. But anyway, that's the fingertip, and there's the actual vein is right there. Right, and that's the artery. Arteries blow out and they explode out from the weak spots and it just happens. Now, and let's look at this. This one was also DNA tested. You see these little wrinkles here? That's fingerprint. That's the skin from a thumb. It was a thumb, and this little piece right here is the piece that's missing. That's what we took off. That's the chunk of skin right under that little tiny flap right there. And underneath there is your fleshy part. And that is where we took the blood out of. But if you look real close, I don't know if you can see this. You see? No, you're probably not going to see it. It's blurry. There's some little tiny holes in there. And that's where the blood passes up through into the skin. There's not much skin, not much blood in the outer layers of skin, but in the inner layers of skin, of flesh, there is a ton of blood. You know, a little goose. This goose, that's a goose. There's no question. You see the feathers on the back of his head? I didn't draw those on there. Those are feathers, and that's his eye. That's a little goose bill. And then he laid dying like this. His neck went over, and when they separated, it snapped. His neck's in the back. You put a little moisture in there. You can see the neck and everything. You can see the bone is at the top like this. That's where the bone sits. All right, that's it. And like this head here, I had presented this for a CAT scan at the University of Texas in Austin, and it ended up with a lawyer and a police calling me for asking some questions. They absolutely are the most arrogant people I've ever seen or run into in my life. This is obviously blood of two different types coming out of the guy's head. This is actually a knit cap. You can slide, you see that right there? You can slide a playing card right up in there. I did the autopsy on this. I worked with Jim Burchell on this, and then not only wouldn't look at it anywhere in the university system, Scott Walter, whatever his name is, on TV said, "Oh, it's just a sandstone head," and then he would not engage. "Oh, I'm a hero. I'm a geologist," some kind of thing he said, and he wouldn't talk. Well, he's got a lot to learn, my friend. And I see him now still talking about things. He's wrong every single time I've seen him. I disagree with him because he's not right. Now you can actually even see the guy's pupil if you look at here. You really got look. It's it. You can actually see the eyes that are in here. And, who breaks the guy's nose apart to carve him? They pay no attention to anything. And it was a terrible, terrible, terrible experience down at the University of Texas, I can tell you that. This is a DNA certified fingertip. Now, if you can see that black pattern, I don't know if you can see it or not. It's been sitting here for years, and it was real black when it first came out of the ground, but I washed it 100 times. Now, if you look at it very carefully, you're going to see the pattern that is the pattern of a distal phalanges. And that's it right there. You see the tip of the finger? All right, now, you see the little bulbous looking thing that's coming off here? That's the apical tuft. That's this right here. All right, you see that? That's the tip of the finger. It's right there. Now that's the ball, as I showed before, it's anchored in there. Now, the knuckle is right there. That is the part that was from the knuckle. And if I showed one little spot that came off, you see here, whoops, you can't see it. Over here. All right. You see the bone ball and the way the muscles wrapped around the bone ball, they come around this way. One side has it, one side is eroded away, as I showed you before. Now on the side that has it, it's this here, this comes over the top, and then it breaks at the abrupt transition. They all do. There's an abrupt transition between tendinous stuff and then muscle fibers. All right, so that's the things I showed you in those big fingertips and the knuckle and that sort of stuff. Now that giant hand I showed you is only right to there. All of these bones and things in the middle of the hand aren't there. It's just the bones in the, well, hold on, over here, look. Let's see if I get up in here. All right. Right here, I believe it's right up to here, or it might only be right, no, it's only right up to here. And all these bones are missing from the front owl. It's just the very bottom of the palm. Right out to here, I believe. It's hard to tell for certain, but and I haven't seen it for 4 or 5 years. But it's not the full palm. Let me put it that way. If you look over here, it's somewhere right about here. If you look at it, you'll see it's right up about here. And then there's parts missing from here and all the fingertips. So of course I found a lot of them, but not everything. It's all on the ground. I got a ton of stuff in the ground. I can see it. I can't dig it up. It's too big. And I hurt my shoulder doing this stuff, and I'm just going to leave it there until somebody gets interested. So once again, I've proven my case in every single possible scientific way, which means every possible legal way. Now this is the ancient bacteria from the ancient DNA extraction kit and so forth, and this is all the details of these 3 specimens, and they all tested to be human mitochondrial DNA 100%, and the negative controls were negative. Here's all this stuff. Now all I know about this is it signifies that these are actually humans, as I allege to Yale and Harvard and all the top universities, and they have refused to acknowledge even, well, Yale said they looked at it and there's nothing here, they can't believe the DNA and all that stuff, but they won't see it in person. So as far as I'm concerned, nobody's really looked at it, which puts them in a very, very, very vulnerable position as a fiduciary, because they should be there to represent the best interests of the students, and their future lives are so dependent upon these people that they've given every bit of trust to, and they're not doing anything to try to do what they should to be sure that they're telling them what is true. It's been presented. There's no question I've proven this case, and in a court of law it'll be seen as true, and I have a feeling it's going to have to end up there, because these students are paying dearly for this. What are they getting? What are they getting? Even all of the research I've done is very, very, very valid, and it's been presented to very, very, very many places, and every one of them has totally ignored it. So all I can say is I have no money involved in these institutions, but if I had paid them, I certainly would want my money back. I would want my money back. I don't know about you, but if they don't want to even take the time to look at what is scientifically valid above anything that they have presented, and obviously legally acceptable. I can't see anybody would look at this and say, oh, the guy's crazy. I would see anybody look at this and say these people are crazy for paying these people for what they're not doing. All right, so I'm just putting out a word of warning. I would jump on this academics very, very quickly, because this is very rapidly going into a serious problem for you people. And that's not my intention. It never was my intention. I put this out to you and said, go do this. You wouldn't do it. I was put in this position. And I don't like it. I'm going to be honest with you. I don't like it. I haven't liked it from the beginning. All I wanted to do was present this and have it looked at for truth and reality, and I will not stand back and say, oh, I don't care, because I do care. It's you that don't care. Hidden Inca tours, and he does fabulous, fabulous research. However, he's stuck with these academics, and he cannot speak against them. He decided he would go and do the same research that I did, go and look into these elongated heads. Nobody's looked at them. Not a single academic has done anything to investigate this. None whatsoever. They do not care. So he took it on himself, just like I took it on myself. So what did he do? He had the academic. He went and had all these very, very sophisticated DNA tests, same as I did, and had all kinds of experts look into this. And I'm sure he had to pay somebody. They don't do these things for free. And then what happened? Let's hear from Brian.
Haplogroup on the maternal side, H2A, which again is Eurasian. So this is the mystery of the elongated skulls of Paracas in Peru that basically no academics are studying. I seem to be the only one, and you, the general public, who are interested in the study of these.
Total disgrace. Last word. All right, I'm going to give you a little background on how dismissed and how against these discoveries academia is. This is Mary Schweitzer. Now, this goes back to 2010, published December 2010. This is a 60 Minutes interview with her where she discovered soft tissues in a dinosaur. Quite by accident, they gave her scraps of bones and she started putting acids on them and so forth. All of a sudden she realized this stuff is getting gooey, and she starts looking at a microscope and there's cells in there. There's blood cells, which is, I understand, is absolutely. And the problem is with her, she's looking in the dust and the dirt. Obviously they're almost gone. Where I'm looking, they are actually gushing blood out of them, as I have shown you. Now, what happened to her? She literally got destroyed. I mean, I'm telling you, I don't care what she says. She thinks she's doing all right. She is not. And I tried to get a hold of her. I got a funny story about that. I don't know if I should tell you or not, but what the hell I'm going to tell you. She's another academic, wouldn't contact me back. Nobody ever did. So I decided, well, I'll call the guy that has her office right down the wall from her. I just can't get a hold of Mary. Could I get a hold of her? He goes down and gets a hold of her. She calls me back, or I she called me back. She just sent me a thing saying, don't ever contact me again. I do all my own stuff only, and leave me alone. Basically, it was kind of abrupt. I understand. My wife said, you're very obnoxious to do that. And I said, well, I'm fed up with these people. Nobody will talk to me. And still nobody's talking to me. Now, they have thrown her out of the nest. Because they actually fired the guy that was working with her, and for saying that he had found this stuff and it was only 3,000 years old. And she's right. It's very, very recent. The KT boundary flood was only, I believe I pegged it somewhere 4,300 and something years ago, according to all of the historic things and all of the mud fossils. And the KT layer is clay, from kaolin clay, from skin, not from an impact. And skin has iridium in it, and that's where the iridium came from. I know that for a fact. So they're starting with 65 million years ago for these dinosaurs. Mary was right. She was right about everything. I tried to help her, but she went her own way, and she's out in the woods now, as far as I can see. Now listen to this.
There's something about dinosaurs that captures the imagination. Giant, mysterious animals that roamed the earth for millions of years, now gone forever. All they've left us are their fossils, the dried out mineral remnants of the creatures they once were, with the organic material that gave them life long gone.
All right, now obviously this is long before I discovered the mud fossils. Well, not long before, actually. In 2012, I knew about mud fossils and about the just gushing blood inside of these things.
Or so everyone always thought. That is until B. rex, a 68-million-year-old Tyrannosaurus rex who was dug up and named by a paleontologist from Montana State University, who, as Leslie Stahl discovered, has an unorthodox approach to dinosaurs, which may be changing the whole dino ballgame.
They won't let it change. Now, I'm gonna cut to the chase on this. Let me, go up and see this thing yourself. If you're that interested, I'm telling you right now, they destroyed it. And I'm gonna show you where she is right now. All right, this is just recent. I'm telling you, she's in trouble with these people. Defying conventional wisdom. She works to transform dinosaur paleontology into a molecular sense. I don't care what they say about me. Paleontologists stare down critics in their hunt for. Well, she's afraid to look into the mud fossils because I'm talking about giant humans, so they throw her right under the bus. Well, I don't care because I have the specimens, and they are the ones that will be in trouble in the end. I can guarantee you that. Now, one last bit of information to share with you. I took a genomics course at Johns Hopkins, and my professor was Stephen Salzberg. And he's a noted DNA specialist with woolly mammoths. He was very, very happy to tell me all about that. We had some nice discussion. It was nice. And then I asked him to take a look at my work and sent it to him at the same time. I said, would you take a look at this, please? And within 8 minutes, I received the nastiest, most arrogant, demeaning reply that you could imagine. That I had no idea what I was doing, that the lab obviously was faulty. Every single thing, I had no clue what I was doing. This is 8 minutes it took him to make this response to 5, 6, 7 years of work. So I don't see much competence there, and I don't see any scientific inquiry whatsoever from Stephen Salzberg at Johns Hopkins. That's my opinion of him. And I would like him to answer to the facts of all of the evidence that I have put forward in a true manner, not just to say to a student, get lost. And that's what happened. And I did. And I told him I would not go silently, and I am not. And this has happened every single time I've ever engaged with anybody in the academic community, and it usually comes out that I am strictly a creationist and I want to prove God. Has nothing to do with that, although I have literally proven God. That is the rub. They hate God. They will not accept God. If you go there, you have to. That is the biggest cult on the face of the planet right now. You have to demean yourself and get on your knees to whatever they tell you to do, and then you have to pay them for them to say that you remembered what they told you to tell them. So, good luck.